T cell. A T cell is a type of lymphocyte which develops in the thymus gland, hence the name, and plays a central role in the immune response. T cells can be distinguished from other lymphocytes by the presence of a T cell receptor on the cell surface. These immune cells originate as precursor cells, derive from bone marrow, and develop into several distinct types of T cells once they have migrated to the thymus gland. T cell differentiation continues even after they have left the thymus. Groups of specific, differentiated T cells have an important role in controlling and shaping the immune response by providing a variety of immune-related functions. One of these functions is immune-mediated cell death, and it is carried out by T cells in several ways. CD8 plus T cells, also known as killer cells, are cytotoxic. This means that they are able to directly kill virus-infected cells as well as cancer cells. CD8 plus T cells are also able to utilize small signaling proteins, known as cytokines, to recruit other cells when mounting an immune response. A different population of T cells, the CD4 plus T cells, function as helper cells. Unlike CD8 plus killer T cells, these CD4 plus helper T cells function by indirectly killing cells identified as foreign. They determine if and how other parts of the immune system respond to a specific, perceived threat. Helper T cells also use cytokine signaling to influence regulatory B cells directly, and other cell populations indirectly. Regulatory T cells are yet another distinct population of these cells that provide the critical mechanism of tolerance, whereby immune cells are able to distinguish invading cells from self, thus preventing immune cells from inappropriately mounting a response against oneself, which would by definition be an autoimmune response. For this reason, these regulatory T cells have also been called suppressor T cells. These same self-tolerant cells are co-opted by cancer cells to prevent the recognition of, and an immune response against, tumor cells. Development. Origin, early development and migration to the thymus. All T cells originate from c-KIT plus SCA1 plus hematopoietic stem cells, HSC, which reside in the bone marrow. In some cases the origin might be the fetal liver during embryonic development. The HSC then differentiate into multipotent progenitors, MPP, which retain the potential to become both myeloid and lymphoid cells. The process of differentiation then proceeds to a common lymphoid progenitor, CLP, which can only differentiate into T, B or NK cells. These CLP cells then migrate via the blood to the thymus, where they engraft. The earliest cells which arrived in the thymus are termed double negative, as they express neither the CD4 nor CD8 coreceptor. The newly arrived CLP cells are CD4, CD8, CD44 plus C.D. 25 KIT plus cells, and are termed early thymic progenitors, ETP, cells. These cells will then undergo a round of division and downregulate c-KIT and are termed DN1 cells. TCR beta selection. At the DN2 stage, CD44 plus C.D. 25 plus, cells upregulate the recombination genes RAG1 and RAG2 and rearrange the TCR beta locus, combining VDJ and constant region genes in an attempt to create a functional TCR beta chain. As the developing thymocyte progresses through to the DN3 stage, CD44 CD25 plus, T cell expresses an invariant alpha chain called pre-T alpha alongside the TCR beta gene. If the rearranged beta chain successfully pairs with the invariant alpha chain, signals are produced which cease rearrangement of the beta chain, and silences the alternate allele. Although these signals require this pre-TCR at the cell surface, they are independent of ligand binding to the pre-TCR. If the pre-TCR forms, then the cell downregulates CD25 and is termed a DN4 cell, CD25 CD44. These cells then undergo a round of proliferation and begin to rearrange TCR alpha locus. Positive selection. Double positive thymocytes, CD4 plus C.D. 8 plus, move deep into the thymic cortex, where they are presented with self-antigens. These self-antigens are expressed by thymic cortical epithelial cells on MHC molecules on the surface of cortical epithelial cells. Only those thymocytes that interact with MHCI or MHC2 will receive a vital survival signal. All that cannot, if they do not interact strongly enough, will die by death by neglect, no survival signal. This process ensures that the selected T cells will have an MHC affinity that can serve useful functions in the body. The cells must be able to interact with MHC and peptide complexes to effect immune responses. The vast majority of developing thymocytes will die during this process. The process of positive selection takes a number of days. A thymocyte's fate is determined during positive selection. Double positive cells, CD4+/C.D8+, that interact well with MHC class II molecules will eventually become CD4+ cells, whereas thymocytes that interact well with MHC class I molecules mature into CD8+ cells. A T cell becomes a CD4+ cell by downregulating expression of its CD8 cell surface receptors. If the cell does not lose its signal, it will continue downregulating CD8 and become a CD4+, single positive cell. This process does not remove thymocytes that may cause autoimmunity. The potentially autoimmune cells are removed by the process of negative selection, which occurs in the thymic medulla, discussed below. Negative selection. Negative selection removes thymocytes that are capable of strongly binding with self-MHC peptides. Thymocytes that survive positive selection migrate towards the boundary of the cortex and medulla in the thymus. While in the medulla, they are again presented with a self-antigen presented on the MHC complex of medullary thymic epithelial cells, MTECs. MTECs must be air plus to properly express self-antigens from all tissues of the body on their MHC class I peptides. Some MTECs are phagocytosed by thymic dendritic cells. This allows for presentation of self-antigens on MHC class II molecules. Positively selected CD4+ cells must interact with MHC class II molecules, thus APCs, which possess MHC class II, must be present for CD4+ T cell negative selection. Thymocytes that interact too strongly with the self-antigen receive an apoptotic signal that leads to cell death. However, some of these cells are selected to become Treg cells. The remaining cells exit the thymus as mature naive T cells, also known as recent thymic emigrants. This process is an important component of central tolerance and serves to prevent the formation of self-reactive T cells that are capable of inducing autoimmune diseases in the host. Beta selection is the first checkpoint, where the T cells that are able to form a functional pre-TCR with an invariant alpha chain and a functional beta chain are allowed to continue development in the thymus. Next, positive selection checks that T cells have successfully rearranged their TCR alpha locus and are capable of recognizing peptide MHC complexes with appropriate affinity. Negative selection in the medulla then obliterates T cells that bind too strongly to self-antigens expressed on MHC molecules. These selection processes allow for tolerance of self by the immune system. Typical T cells that leave the thymus, via the corticomedullary junction, are self-restricted, self-tolerant, and single-positive. Thymic output. About 98% of thymocytes die during the development processes in the thymus by failing either positive selection or negative selection, whereas the other 2% survive and leave the thymus to become mature immunocompetent T cells. The thymus contributes fewer cells as a person ages. As the thymus shrinks by about 3% a year throughout middle age, a corresponding fall in the thymic production of naive T cells occurs, leaving peripheral T cell expansion and regeneration to play a greater role in protecting older people. Types of T cell. T cells are grouped into a series of subsets based on their function. CD4 and CD8 T cells are selected in the thymus, but undergo further differentiation in the periphery to specialized cells which have different functions. T cell subsets were initially defined by function, but also have associated gene or protein expression patterns. Conventional adaptive T cells. Helper CD4+ T cells. T helper cells, TH cells, assist other lymphocytes, including maturation of B cells into plasma cells and memory B cells, and activation of cytotoxic T cells and macrophages. These cells are also known as CD4+ T cells as they express the CD4 on their surfaces. Helper T cells become activated when they are presented with peptide antigens by MHC class II molecules, which are expressed on the surface of antigen-presenting cells, APCs. Once activated, they divide rapidly and secrete cytokines that regulate or assist the immune response. These cells can differentiate into one of several subtypes, which have different roles. Cytokines direct T cells into particular subtypes. Cytotoxic CD8+ T cells. Cytotoxic T cells, TC cells, CTLs, T killer cells, killer T cells, destroy virus-infected cells and tumor cells, and are also implicated in transplant rejection. These cells are defined by the expression of CD8+ on the cell surface. These cells recognize their targets by binding to short peptides, 811A, associated with MHC class I molecules, present on the surface of all nucleated cells. CD8 plus T cells also produce the key cytokines IL-2 and IFN-gamma, which influence the effector functions of other cells, in particular macrophages and NK cells. Memory T cells. Antigen-naive T cells expand and differentiate into memory and effector T cells after they encounter their cognate antigen within the context of an MHC molecule on the surface of a professional antigen-presenting cell, e.g. a dendritic cell. Appropriate co-stimulation must be present at the time of antigen encounter for this process to occur. Historically, memory T cells were thought to belong to either the effector or central memory subtypes, each with their own distinguishing set of cell surface markers, see below. Subsequently, numerous new populations of memory T cells were discovered, including tissue-resident memory T, TRM, cells, stem memory TSCM cells, and virtual memory T cells. The single unifying theme for all memory T cell subtypes is that they are long-lived and can quickly expand to large numbers of effector T cells upon re-exposure to their cognate antigen. By this mechanism, they provide the immune system with memory against previously encountered pathogens. Memory T cells may be either CD4+ or CD8+ and usually express CD45-Rho. Memory T cell subtypes. Regulatory CD4+ T cells. Regulatory T cells are crucial for the maintenance of immunological tolerance. Their major role is to shut down T cell-mediated immunity toward the end of an immune reaction and to suppress autoreactive T cells that escape the process of negative selection in the thymus. Two major classes of CD4+ Treg cells have been described, FOXP3+ Treg cells and FOXP3- Treg cells. Regulatory T cells can develop either during normal development in the thymus, and are then known as thymic Treg cells, or can be induced peripherally and are called peripherally derived Treg cells. These two subsets were previously called naturally occurring, and adaptive or induced, respectively. Both subsets require the expression of the transcription factor FOXP3, which can be used to identify the cells. Mutations of the FOXP3 gene can prevent regulatory T cell development, causing the fatal autoimmune disease IPEX. Several other types of T cell have suppressive activity, but do not express FOXP3. These include TR1 cells and TH3 cells, which are thought to originate during an immune response and act by producing suppressive molecules. TR1 cells are associated with IL-10, and TH3 cells are associated with TGF-beta. Recently, Treg17 cells have been added to this list. Innate-like T cells. Natural killer T cell. Natural killer T cells, NK T cells, not to be confused with natural killer cells of the innate immune system, bridge the adaptive immune system with the innate immune system. Unlike conventional T cells that recognize peptide antigens presented by major histocompatibility complex, MHC, molecules, NK T cells recognize glycolipid antigen presented by CD1D. Once activated, these cells can perform functions ascribed to both Th and Tc cells, i.e., cytokine production and release of cytolytic/cell-killing molecules. They are also able to recognize and eliminate some tumor cells and cells infected with herpes viruses. Mucosal-associated invariant. MATE cells display innate, effector-like qualities. In humans, MATE cells are found in the blood, liver, lungs, and mucosa, defending against microbial activity and infection. The MHC class I-like protein, MR1, is responsible for presenting bacterially produced vitamin B metabolites to MATE cells. After the presentation of foreign antigen by MR1, MATE cell secretes pro-inflammatory cytokines and are capable of lysing bacterially infected cells. MATE cells can also be activated through MR1 independent signaling. In addition to possessing innate-like functions, this T cell subset supports the adaptive immune response and has a memory-like phenotype. Furthermore, MATE cells are thought to play a role in autoimmune diseases, such as multiple sclerosis, arthritis and inflammatory bowel disease, although definitive evidence is yet to be published. Gamma-delta T cells. Gamma-delta T cells, gamma-delta T cells, represent a small subset of T cells which possess a gamma-delta TCR rather than the alpha-beta TCR on the cell surface. The majority of T cells express alpha-beta TCR chains. This group of T cells is much less common in humans and mice, about 2% of total T cells, and are found mostly in the gut mucosa, within a population of intraepithelial lymphocytes. In rabbits, sheep, and chickens, the number of gamma delta T cells can be as high as 60% of total T cells. The antigenic molecules that activate gamma delta T cells are still mostly unknown. However, gamma delta T cells are not MHC restricted and seem to be able to recognize whole proteins rather than requiring peptides to be presented by MHC molecules on APCs. Some murine gamma delta T cells recognize MHC classed molecules. Human gamma delta T cells, which use the V gamma 9 and V delta 2 gene fragments, constitute the major gamma delta T cell population in peripheral blood, and are unique in that they specifically and rapidly respond to a set of non-peptidic phosphorylated isoprenoid precursors, collectively named phosphoantigens, which are produced by virtually all living cells. The most common phosphoantigens from animal and human cells, including cancer cells, are isopentenyl pyrophosphate, IPP, and its isomer dimethylyl pyrophosphate, DMPP. Many microbes produce the highly active compound hydroxide MAP, HMBPP, and corresponding mononucleotide conjugates, in addition to IPP and MAP. Plant cells produce both types of phosphoantigens. Drugs activating human V gamma 9/V delta 2 T cells comprise synthetic phosphoantigens and aminobisephosphonates, which upregulate endogenous IPP/MAP. Activation of CD4 plus T cells occurs through the simultaneous engagement of the T cell receptor and a co-stimulatory molecule, like CD28, or ICOs, on the T cell by the major histocompatibility complex, MHC, peptide and co-stimulatory molecules on the APC. Both are required for production of an effective immune response. In the absence of co-stimulation, T cell receptor signaling alone results in anergy. The signaling pathways downstream from co-stimulatory molecules usually engages the PEC pathway, generating PIP3 at the plasma membrane and recruiting pH domain containing signaling molecules like PDK1 that are essential for the activation of PKC theta, and eventual IL-2 production. Optimal CD8 plus T cell response relies on CD4 plus signaling. CD4 plus cells are useful in the initial antigenic activation of naive CD8 T cells, and sustaining memory CD8 plus T cells in the aftermath of an acute infection. Therefore, activation of CD4 plus T cells can be beneficial to the action of CD8 plus T cells. The first signal is provided by binding of the T cell receptor to its cognate peptide presented on MHC on an APC. MHC is restricted to so-called professional antigen-presenting cells, like dendritic cells, B cells, and macrophages, to name a few. The peptides presented to CD8 plus T cells by MHC class I molecules are 8 to 13 amino acids in length. The peptides presented to CD4 plus cells by MHC class II molecules are longer, usually 12 to 25 amino acids in length, as the ends of the binding cleft of the MHC class II molecule are open. The second signal comes from co-stimulation, in which surface receptors on the APC are induced by a relatively small number of stimuli, usually products of pathogens, but sometimes breakdown products of cells, such as necrotic bodies or heat shock proteins. The only co-stimulatory receptor expressed constitutively by naive T cells is CD28, so co-stimulation for these cells comes from the CD80 and CD86 proteins, which together constitute the B7 protein, B7.1 and B7.2, respectively, on the APC. Other receptors are expressed upon activation of the T cell, such as OX40 and ICOs, but these largely depend upon CD28 for their expression. The second signal licenses the T cell to respond to an antigen. Without it, the T cell becomes anergic, and it becomes more difficult for it to activate in future. This mechanism prevents inappropriate responses to self, as self-peptides will not usually be presented with suitable co-stimulation. Once a T cell has been appropriately activated, i.e. has received signal 1 and signal 2, it alters its cell surface expression of a variety of proteins. Markers of T cell activation include CD69, CD71 and CD25, also a marker for Treg cells, and HLA drive, a marker of human T cell activation. CTLA4 expression is also upregulated on activated T cells, which in turn outcompetes CD28 for binding to the B7 proteins. This is a checkpoint mechanism to prevent overactivation of the T cell. Activated T cells also change their cell surface glycosylation profile. The T cell receptor exists as a complex of several proteins. The actual T-cell receptor is composed of two separate peptide chains, which are produced from the independent T-cell receptor alpha and beta, TCR alpha and TCR beta, genes. The other proteins in the complex are the CD3 proteins, CD3 epsilon gamma and CD3 epsilon delta heterodimers and, most important, a CD3 zeta homodimer, which has a total of 6 atom motifs. The atom motifs on the CD3 zeta can be phosphorylated by LCK and in turn recruit ZAP70. LCK and or ZAP70 can also phosphorylate the tyrosines on many other molecules, not least CD28, LOD and SLP76, which allows the aggregation of signaling complexes around these proteins. Phosphorylated LOD recruits SLP76 to the membrane, where it can then bring in PLC gamma, VAV1, it can potentially PEC. PLC gamma cleaves Pi4,5P2 on the inner leaflet of the membrane to create the active intermediaries diacylglycerol, DAG, inositol 1,4,5 trisphosphate, IP3. PEC also acts on PIP2, phosphorylating it to produce phosphatidylinositol 3,4,5 trisphosphate, PIP3. DAG binds and activates some PKCs. Most important in T cells is PKC theta, critical for activating the transcription factors NF-kappa B and AP1. IP3 is released from the membrane by PLC gamma and diffuses rapidly to activate calcium channel receptors on the ER, which induces the release of calcium into the cytosol. Low calcium in the endoplasmic reticulum causes STIM1 clustering on the ER membrane and leads to activation of cell membrane crack channels that allows additional calcium to flow into the cytosol from the extracellular space. This aggregated cytosolic calcium binds calmodulin, which can then activate calcineurin. Calcineurin, in turn, activates NFAT, which then translocates to the nucleus. NFAT is a transcription factor that activates the transcription of a pleiotropic set of genes, most notable, IL-2 cytokine that promotes long-term proliferation of activated T cells. PLC gamma can also initiate the NF-kappa B pathway. DAG activates PKC theta, which then phosphorylates KARMA1, causing it to unfold and function as a scaffold. The cytosolic domains bind an adapter BCL10 via CARD, caspase activation and recruitment domains that then binds TRAF6, which is ubiquitinated at K63.513 to 523. This form of ubiquitination does not lead to degradation of target proteins. Rather, it serves to recruit NEMO, IKK alpha and beta, and TAB12/TAC1. TAC1 phosphorylates IKK beta, which then phosphorylates I-kappa B, allowing for K48 ubiquitination, leads to proteasomal degradation. RelA and P50 can then enter the nucleus and bind the NF-kappa B response element. This, coupled with NFAT signaling, allows for complete activation of the IL-2 gene. While in most cases activation is dependent on TCR recognition of antigen, alternative pathways for activation have been described. For example, cytotoxic T cells have been shown to become activated when targeted by other CD8 T cells, leading to tolerization of the latter. In spring 2014, the T-cell activation in space (TCAS) experiment was launched to the International Space Station on the SpaceX CRS-3 mission to study how deficiencies in the human immune system are affected by a microgravity environment. T-cell activation is modulated by reactive oxygen species. Antigen discrimination. A unique feature of T cells is their ability to discriminate between healthy and abnormal, for example infected or cancerous, cells in the body. Healthy cells typically express a large number of self-derived PMHC on their cell surface, and although the T-cell antigen receptor can interact with at least a subset of these self-PMHC, the T-cell generally ignores these healthy cells. However, when these very same cells contain even minute quantities of pathogen-derived PMHC, T-cells are able to become activated and initiate immune responses. The ability of T cells to ignore healthy cells but respond when these same cells contain pathogen, or cancer-derived PMHC is known as antigen discrimination. The molecular mechanisms that underlie this process are controversial. Clinical significance. Deficiency. Causes of T-cell deficiency include lymphocytopenia of T cells and/or defects on function of individual T cells. Complete insufficiency of T-cell function can result from hereditary conditions such as severe combined immunodeficiency, SCID, Omenn syndrome, and cartilage hair hypoplasia. Causes of partial insufficiencies of T-cell function include acquired immune deficiency syndrome, AIDS, and hereditary conditions such as DiGeorge syndrome, DGS, chromosomal breakage syndromes, CBSs, and B-cell and T-cell combined disorders such as ataxia-telangiectasia, AT, and Wiskott-Aldrich syndrome, WAS. The main pathogens of concern in T-cell deficiencies are intracellular pathogens, including herpes simplex virus, mycobacterium and listeria. Also, fungal infections are also more common and severe in T-cell deficiencies. Cancer. Cancer of T-cells is termed T-cell lymphoma, and accounts for perhaps 1 in 10 cases of non-Hodgkin lymphoma. The main forms of T-cell lymphoma are exhaustion. T-cell exhaustion is a state of dysfunctional T-cells. It is characterized by progressive loss of function, changes in transcriptional profiles and sustained expression of inhibitory receptors. At first cells lose their ability to produce IL-2 and TNF-alpha followed by the loss of hyperproliferative capacity and cytotoxic potential, eventually leading to their deletion. Exhausted T-cells typically indicate higher levels of CD43, CD69 and inhibitory receptors combined with lower expression of CD62L and CD127. Exhaustion can develop during chronic infections, sepsis and cancer. Exhausted T-cells preserve their functional exhaustion even after repeated antigen exposure. During chronic infection and sepsis, T-cell exhaustion can be triggered by several factors like persistent antigen exposure and lack of CD4 T-cell help. Antigen exposure also has effect on the course of exhaustion because longer exposure time and higher viral load increases the severity of T-cell exhaustion. At least 2 to 4 weeks exposure is needed to establish exhaustion. Another factor able to induce exhaustion are inhibitory receptors including programmed cell death protein 1, PD1, CTLA4, T-cell membrane protein 3, TIM3, and lymphocyte activation gene 3 protein, LAG3. Soluble molecules such as cytokines IL-10 or TGF-beta are also able to trigger exhaustion. Last known factors that can play a role in T-cell exhaustion are regulatory cells. Treg cells can be a source of IL-10 and TGF-beta and therefore they can play a role in T-cell exhaustion. Furthermore, T-cell exhaustion is reverted after depletion of Treg cells and blockade of PD1. T-cell exhaustion can also occur during sepsis as a result of cytokine storm. Later after the initial septic encounter, anti-inflammatory cytokines and pro-apoptotic proteins take over to protect the body from damage. Sepsis also carries high antigen load and inflammation. In this stage of sepsis, T-cell exhaustion increases. Currently there are studies aiming to utilize inhibitory receptor blockades in treatment of sepsis. During transplantation. While during infection T-cell exhaustion can develop following persistent antigen exposure after graft transplant, similar situation arises with alloantigen presence. It was shown that T-cell response diminishes over time after kidney transplant. These data suggest T-cell exhaustion plays an important role in tolerance of a graft mainly by depletion of alloreactive CD8 T-cells. Several studies showed positive effect of chronic infection on graft acceptance and its long-term survival mediated partly by T-cell exhaustion. It was also shown that recipient T-cell exhaustion provides sufficient conditions for NK cell transfer. While there are data showing that induction of T-cell exhaustion can be beneficial for transplantation, it also carries disadvantages, among which can be counted increased number of infections and the risk of tumor development. During cancer, T-cell exhaustion plays a role in tumor protection. According to research, some cancer-associated cells as well as tumor cells themselves can actively induce T-cell exhaustion at the site of tumor. T-cell exhaustion can also play a role in cancer relapses, as was shown on leukemia. Some study even suggested that it is possible to predict relapse of leukemia based on expression of inhibitory receptors PD1 and TIM3 by T-cells. In recent years there is a lot of experiments in clinical trials with immune checkpoint blockers in cancer therapy. Some of them were approved as valid therapies and are now used in clinics. Inhibitory receptors targeted by those medical procedures are vital in T-cell exhaustion, and blocking them can reverse these changes. See also immunosenescence changes. See also immunosenescence changes. See also immunosenescence changes. See also immunosenescence